Let's now move to the fourth talk of this track. And it's my pleasure to introduce you to Dr. Adel Mustafa from the Department of Pathology, Tumor Immunopathology Laboratory of the Erasmus University Medical Center in Rotterdam. The topic of her talk is the immune microenvironment of pancreatic cancer.
Hello, good afternoon from the Netherlands. I'm Adel Mustafa, and I'm an assistant professor and the leader of the genomics facility at the Tumor Immune Pathology in Erasmus Medical Center. Today I will talk about, as the title indicates, I will talk about immune infiltration and how the microenvironment of pancreatic cancer may influence the survival of the patients. But before I start, I'd like to first give my thanks to NanoString Company for arranging this virtual oncology meeting and for giving me the opportunity to talk about our work in the lab. I'd also like to thank the chair lady who introduced me a bit earlier. So let me start by refreshing your memories about the pancreas. Pancreas is part of our digestive system. It is in the abdomen just behind the stomach, and it's two-in-one organ. So it functions as a gland, and it has two functions basically: endocrine function that regulates blood sugar, and exocrine function, mainly by producing enzymes to digest food. The type of pancreatic cancer depends on the cell's origin. If cancer starts from islet cells, these are the cells that they are responsible to produce insulin, endocrine cells, then the type of cancer is going to be neuroendocrine or pancreatic neuroendocrine tumor, and it presents almost 10% of pancreatic cancer. We lost some famous people for this type of cancer. We lost Franklin and we lost Steve Jobs for this type of cancer. If pancreatic cancer originated from acinar cells, we refer to it as pancreatic ductal adenocarcinoma, and this is a complete different type of tumor. We lost, unfortunately, some famous people, Pavarotti, and recently, this year, we lost Irfan Khan for this type of tumor. Pancreatic cancer, especially pancreatic ductal adenocarcinoma, I refer to it in my talk as pancreatic cancer, but what I mean is pancreatic ductal adenocarcinoma, which is the most common type of pancreatic cancer, and it represents more than 90% of pancreatic cancer. Unfortunately, it's a very bad disease. It's expected to be the leading cause of cancer-related death in 2030. It's painful disease because the tumor cells invade nerves. They also invade lymphatics and veins, and they metastasize. The 5-year survival rate is 5%, and most patients die within 2 years. The reason for that, because it's an asymptomatic disease. Until the patient develops some symptoms and they come to the clinic to be checked, the cancer is already there, and it's in a lot of cases, it's already spread. At a histological level, the cancer, pancreatic ductal adenocarcinoma, they are the granular or ductal differentiation. So it's the epithelial neoplasm of granular ductal differentiation. It's a highly heterogeneous disease. It's characterized by remarkable dysplasia or fibroblasts. In some cases, dysplasia can represent 50% of the tumor or even more. They have a very poor angiogenesis or blood supply. The dysplasia is a very important component of pancreatic cancer, and it mainly composed of extracellular matrix proteins, fibroblasts, a lot of fibroblasts, various types of fibroblasts, pancreatic stellate cells, and immune cells. Immune cells or tumor-infiltrated lymphocytes, recently, since 5-7 years ago, they started to have a huge highlight because of the importance of these immune cells as a part of the tumor. It's usually the balance between the tumor suppressor and the tumor progressor. It's a balance in the immune cells that drive the tumor either towards suppression or progression. So we usually refer to them as good and bad players. Not per se, it's the balance between the good and bad players is the most important thing. They can predict the response to therapy, overall survival, progression-free survival. It's very important to know not only the amount of infiltration cells, not only the type of infiltration cells, but also their location and their ratio relative to each other. Understanding the immune system may open therapeutic options like we see with immune checkpoint inhibitors. Now, previously, our group has published and studied the immune component of pancreatic cancer. We have, I'm referring to one of the publications, and in this publication we did by immunohistochemistry, we stained for several types of T cells, like CD8, CD4 T regulatory cells marked by FOXP3, also some immune checkpoint inhibitors. And what we concluded in that article, pancreatic cancer or pancreatic ductal adenocarcinoma to be specific, is low in CD8 T cell infiltration and high in immune suppressive myeloid cells. We also show an evidence that the CD8 to FOXP3 ratio, the high CD8 to FOXP3 ratio, is associated with favorable outcome, like Kaplan-Meier shows here. If we look at the literature and we search for pancreatic cancer together with immune microenvironment or microenvironment, we see a number of publications has increased in the last 5 to 7 years, and that is in relation with the immune checkpoint inhibitors and the immune therapy that's been introduced to treat cancer patients in general. So there is a demand and there is a huge need to understand the microenvironment and specifically the immune microenvironment, not only in pancreatic cancer, but also in other types of diseases. And that is one of our focus in our group, understanding the immune microenvironment of pancreatic ductal adenocarcinoma. Now, several publications, I'm referring to few here, they hypothesized and suggest the relation between tumor-associated macrophages and fibroblasts, which is the dysplasia. This is where mainly we see a lot of infiltration of immune cells. Some inhibit and some suppress tumor cells. There is recently a review and a meta-analysis trying to understand the prognostic value of tumor infiltration, and trying to understand, other publications are highlighting the importance of understanding the landscape of immune cell infiltration to predict the clinical outcome and to have an implication in pancreatic ductal adenocarcinoma. I'm listing here a few examples, the recent publications. However, what I like to highlight is most of these publications are focusing on T cells. So the highlights of these publications is about the infiltration of T cells in pancreatic cancer. They predict better survival, especially CD8 associated with better outcome. T regulatory cells or FOXP3 T cells are associated with poor outcome. But as you see, most of these publications are about T cells. And they recommend that other types of cells, like for example CD20, which are B cells or natural killer cells, should be studied. The rationale of what I'm presenting today is, unfortunately, most of pancreatic cancer patients, they die within the first couple of years, as I told you earlier. However, there is a very small subgroup of patients, they survive for many more years. So there are factors to determine the long-term survivorship, mainly are known. And we hypothesize that the tumor microenvironment and the immune cell infiltration may be the key to determine the long-term survival in pancreatic ductal adenocarcinoma. So based on this hypothesis, our aim and the aim of what I'm presenting today is to reveal the immune and the microenvironmental pathway that may determine long-term survival in pancreatic cancer patients. To achieve our aim, we went back to the archive. We have more than 20, we collected a big archive, more than 25 years. And we went to all these patients' material and their clinical information to know their clinical evaluation and treatment. And we selected the maximum number of survivals. So we have 10 patients who survived for 5 years or longer. And we compared that to 10 patients who survived for 6 months or shorter. And that was our definition for long and short term survival. And the first thing we did is we immune profiled them using an encounter facility, nanostring technology. And afterwards, we selected a subgroup of them to be studied in the geomics DSP, and I'll come to that in details later. Our workflow, we selected in this case, in the first part of the study, we selected fresh frozen material. We enrich for tumor tissue, so we sectioned the tissue samples, and a pathologist, an experienced pathologist, would study them. And then we enrich for tumor tissue, meaning we enrich for tumor cells, including dysplasia or stroma, that's part of the tumor. Then we did RNA isolation. We checked the quality, and we used 200 nanograms per reaction, which includes 5 microliters of RNA, good quality RNA, but fresh frozen material usually gives good quality RNA. And then we used the immune profile panel from NanoString. We hybridized for 17 hours. We scanned 490 fields of view, and we used the Insolver advanced analysis module to analyze our samples. And then we validated using the geomics DSP. Now, a technical detail, I'm not aware of how familiar you are with the nanostring technology and the principle of the work, but just for a short notice, we used the PanCancer Immune Profile panel, including 770 genes representing the innate and the adaptive immune system, 40 housekeeping genes. There are 24 different immune cell types and several pathways. And the advantage of using the panels from NanoString is you hybridize RNA immediately with 2 props. So you capture 100 base pair of the RNA immediately without enzymes, using 2 props, 50 base pair each. And each prop at the end has a flag or a reporter prop of 6 fluorescent dyes to present various genes. So it will give a count of the genes of your interest. Here's a volcano plot of the differentially expressed genes. So in this side, in the right side, there are the genes that they are overexpressed or upregulated in the long-term survivals. And here are in the green direction, there are the genes that they are overexpressed in the short-term survivals. Please notice that the y-axis represents the log10 p-value, and the x-axis represents the log2 fold of change. So 1 here is 2 in linear form. So we have more than 14 differentially expressed genes at a p-value of 0.01 and a fold of a change of 1.5 in both directions. If we take these 14 genes, in order to distinguish between the 2 groups, remember we have 10 patients per group, then there is a good separation. We did principal component analysis, and in gray you see long-term survivals, that they are separated from the short-term survivals. Of course, they overlap a little bit, but the overlapping is not that big. So we are very happy with these results. And then you can take the normalized data, and you can create heat maps, or you can study. For example, I'm very much interested in the most differentially expressed gene between both groups. But you are allowed to do all of it. based on your taste. What I'm also very interested in is knowing the differentially expressed immune cell types. So how can we utilize gene expression results? By giving cell markers to identify immune cells. I'm giving an example of a B cell, of the definition of B cells. We used 4 different markers. Then we calculate the correlation coefficient between these 4 markers. And for us, we define them based on, or we trust cell markers that they have a correlation coefficient higher than 0.6. Meaning, like these cell markers behave in the same way across all the samples, and then you can trust them to give you a score for cells. Now, if we look at the infiltrative cells defined by CD45, they are not different between long and short cell life. But if we look at the B cell score, then it's a significant different score, which represents a higher score in long-term survivals as compared to short. Again, the definition of the B cells in this case were represented by 4 markers. This is CD20, CR2, CD22, and CD19. And this attracts our attention because actually the role of B cells in pancreatic cancer remains controversial. It is suggested until now that it has a role of immune suppression. There are 3 publications were published in 2016. They suggest or they indicate that B cells fuel pancreatic cancer, pancreatic ductal adenocarcinoma, and they promote pancreatic tumorigenesis. However, a more recent publication in 2019 suggests the opposite. Please remember that these publications are limited with the number of patients they used or samples of patients they used. They also are limited by the technique. Most of these publications are based on immunohistochemistry, and a lot of them are based on using animal models. We know that animal models, they don't represent, especially when we talk about immune cells or immune system, they don't represent the human situation. But we have opposite information in the literature. Our results shows that B cells infiltrate more in the long-term survivals as compared to the short-term survivals. And that raises a lot of questions. I'm going to address 2 of them today. One of the questions is the location. Where do they infiltrate? I introduced the tumor as a heterogeneous tumor, so I'd like to know where exactly they infiltrate and what are the other types of cells that they interact with. And to answer these questions, NanoString has developed the GeoMx Digital Spatial Profiler platform. In my opinion, it is a fantastic platform to answer our questions. So now we take a tissue sample and we stain the tissue sample with morphological markers. So we distinguish between tumor cells. In this case, we distinguish them with pancytokeratin marker in yellow, and we distinguish alpha smooth muscle actin in green, and any tumor cells which are positive for CD45, they are in red. And what we did previously using the Encounter platform is one sample, we took a section, we isolated one RNA sample, and then we did one measurement. But now with the DSP, one sample can be as many as we want. We are focusing on pancytokeratin, CD45, and alpha smooth muscle actin. In other words, using this technology, you are able to connect biology to morphology or expression to morphology. How does it work? NanoString developed immune oncology module in which it includes the core module to distinguish the most common types of immune cells. As you can see, CD20, CD3, CD4, CD45, and so on and so forth. And then they are coupled with other modules based on your question, like drug, immune oncology drug target modules, or activation status, suppressive status, and across all types of tumor cells. These are antibodies. And these antibodies are conjugated with oligo. That oligo is UV cleavable. So in practice, what we do is we take a tissue section of 4 or 5 micrometer of paraffin-embedded section, and we stain that section with our morphological markers, pancytokeratin, alpha-smooth muscle actin, and CD45. And then we hybridize this section after their morphological markers with the cocktail of these antibodies. We leave it overnight, and next day we are able to distinguish based on the segments. We are able to profile each one of our segments separately. And here is a real example. This is one of the samples that we measured. And each one of these boxes, these are 12 boxes, ROIs, or region of interest. In each one of these regions of interest, you see a circle, and that represents, for example, circle number 7 is full of green color. Green is alpha-smooth muscle actin. Please notice that it is high. Area number 7 is high in alpha-smooth muscle actin. It is very low in pancytokeratin, and it is very low in CD45. And now we profile them. I'll go to my target. This is an example of a long-term survival sample in which I chose this area. This area is filled with with immune cells. And when we look at the profile of this area, they were very high in CD20, which is a marker for B cells. I forgot to tell you that the data can be normalized in different ways. What I'm presenting here, the data were normalized using positive controls and housekeeping genes. So, area number 8 in a long-term survival sample was filled with or had a high expression counts of CD20. And here is another example of a short-term survival in which I chose exactly the same morphological area. This area is filled with immune cells. It is low in alpha-smooth muscle actin, and it is low in pancytokeratin. This is area number 10. And if we look at the expression or the counts, this area is filled also with CD20, which is a marker of B cells. If we put these data together and we notice the value of the y-axis, which is the count, these two areas, which is filled with immune cells, are filled with B cells as well, but in a complete different range. Long-term survival showed a range of 5,000 counts, and short-term survival showed a count of 600. Now, this is one sample versus one sample. If you take 6 versus 6 and you put all the information together, the long-term survivals are much higher in infiltration with B cells as compared to short-term survivals. And I'm here presenting areas that they were filled with immune cells. I'm not presenting pancytokeratin. In order to show it to you in a different way, these are the areas of alpha-smooth muscle actin, so the dysplasia in pancreatic cancer, in which the area in the right are overexpressed in short, and the genes or the markers in them, these are not genes, these are proteins, in the left are overexpressed in long. And please notice with me that CD20 is overexpressed with a good p-value and a very high fold of a change, twice as much in long-term survival as short-term survival. And they are in a close proximity with T cells, CD4, CD3, CD11C, and CD68, which is macrophages and monocytes. If we take the areas of pancytokeratin, which represent the tumor cells, then we don't reach significant results. In the long-term survival, we see the same trend, but it is not significant. However, we see a lot of significance in FOXP3, T regulatory cells, and immune suppressive cells in the tumor cells themselves. So, putting this information together, we saw that B cells infiltrated in the stroma of long-term survival, and they are in close distance with CD4, CD8, CD68, CD14, and CD11C cells. B cells had gained a lot of interest at the beginning of 2020. In February 2020, 3 publications came in Nature almost in the same week. And all of them were referring to the important role of B cells in the long-term survival or in the good prognosis of various types of cancer, like melanoma, sarcoma, and others. But we are the first to show this role in pancreatic cancer. So I'm now in the conclusions, and I'd like to conclude with you to remind you that the immune infiltration is one of the keys that determine the survival in pancreatic ductal adenocarcinoma. They are infiltrated more in long-term survivals, meaning patients who survived 5 years and longer, as compared to patients who survived 6 months and shorter. And B cells infiltration is associated with T cells, M2 cells, and DCs, dendritic cells. The high infiltration of T regulatory cells was monitored in desmoplastic tissue in the ductal tumor cells of ductal tumor tissue of pancreatic ductal adenocarcinoma. We have a lot of more questions, and we actually have clinical data to answer our questions, like to connect the infiltration with the clinical parameters we have, or to study the pancytokeratin or the tumor cells a little bit more in details to know what types of cells infiltrate in the short-term survival in that case. But with that, I would like to conclude my presentation, and I hope I convinced you that B cells play an important role in determining the survivalship of pancreatic ductal adenocarcinoma. I'd like to thank people who participate in the study from the TIP lab or the Tumor Immune Pathology lab at Erasmus Medical Center. And that lab is led by Professor Van Eyck. I'd also like to thank the bioinformatics team from NanoString that helped me to analyze the data generated by Genomics DSP. And I very much appreciate the involvement of the patients, the donation of their tissue that enabled us to study their tissue. And I'd like to thank Support Caspar for the financial support. And I will be very happy to answer your questions.
Thank you very much, Dr. Mustafa, for the nice talk. I have a few questions for you too. Can you please expand on how one can identify immune cell subset using gene expression?
Expression of genes. And actually, I received this question quite a lot. I'd like to go back to my slides and I show you the example of B cells, identification of B cells. So we use different gene markers to identify an immune cell type. And actually, for pancreatic cancer, there is a publication in preparation right now. We use the Cancer Genome Atlas data, and we calculate all the definition of cells by calculating the correlation coefficient between the genes. So using public data, which is from the Cancer Genome Atlas, we calculated the correlation coefficient between the different types of cell markers. And we trusted those that they have 0.6 and higher to define the types of cells. In that way, we are able to identify various types of cells in a very confident way. So the default of NanoString or InSolver advanced analysis, the default gives various types of cells, but we were able to increase that. For example, NanoString gives the B cells. We were able to increase it to plasma B cells, regulatory B cells, and memory B cells in our data set. We also do the same for T cells. You do the same for dendritic cells, for any type of cells. You distinguish, or from the literature, you know the markers, the gene markers, to identify types of cells, and then you check the correlation coefficient between the genes in order to determine if you trust them in your data set or not. That's how you define immune cells using gene expressions. There are other softwares that they do that. One of the famous one is cell sort or super sort. So it's a very well-known technique.
Thank you. Since in your study, you have used both the bulk technology platform as well as the tissue imaging platform, which kind of advice would you give to researchers about which one to use when the goal of your research is to discover difference between two groups of interest?
Two groups. It is based on your personal taste and the time, money, and effort, number of samples that you have in the study the scientists want to do, I think, or the scientists want to perform. The way I chose is to profile the samples using Encounter and then validating using geomics, digital spatial profile. The reason for that is because I think it's a little bit faster and cost effective if you do it this way. You will study more samples with the Encounter, and then you can validate your results using a smaller cohort of patients or of samples. However, you can do the same. You can do also the discovery and the validation using geomics DSP. It depends on the samples, on the number of samples included in the study. The DSP will allow you to study the samples in much more details as compared to the Encounter. The Encounter is usually, I refer to it as a soup. So you isolate RNA and from the whole sample, and then you measure the expression of cells, or you measure the expression of genes, not knowing what give rises to these expressions. In the DSP, no, you know exactly where you are located in the tumor. You study the population that you want to study it, and you can do that at two levels. You can do that at the protein level, using antibodies conjugated to oligos, or you can do that also at the RNA level, which I think is a fantastic method to study secreted cytokines. I hope.
Thank you. What is the function of B cells in pancreatic cancer?
Very good question. Our data show that they are higher in patients who survive longer as compared to patients who survive shorter. It's a relative expression. So I'm assuming that I'm confident that the B cells in pancreatic cancer, they play a good role. They actually suppress the tumor. However, we still need to validate, or not to validate, to discover the factors that facilitate B cells to infiltrate to the tumor cells, to the tumor areas in long-term survival. And the function of B cells is still not known in pancreatic cancer. We are working on it, and we are studying samples in much more details. But I cannot really give a precise answer for that question.
Thank you very much, Dr. Mustafa, for the presentation and the discussion.
So at the end of this presentation, I hope I convinced you that B cells play an important role in the long-term survivalship in pancreatic ductal adenocarcinoma. But I hope I bring a little bit a bigger message, which is the importance of studying tumor microenvironment. Again, I'm concentrating here in pancreatic cancer, but we study more tumors. Tumor microenvironment is gaining a lot of attention lately. And the reason for that is because it may open new therapeutic options for patients who suffer from this bad disease. So I think the international effort that we all put together in understanding the tumor microenvironment, whether it's in pancreatic cancer or other types of cancers, it will be great to put all of this effort together and to collaborate, to connect our databases, so to identify or to discover more about the tumor microenvironment. The more we understand the tumor in general, the more we are going to be successful in winning the battle against it. With that, I'd like to conclude. And once again, I'd like to thank the Tumor Immune Pathology Lab at Erasmus Medical Center. And I'd like to tell you that we have the Geomics DSP. We installed it just before Corona era, and I cannot wait to start working with it. I also would like to thank NanoString for hosting and for organizing this virtual conference. And if you have any question, I'd be happy to answer them at any time.